Hi, this is Noe, and I will give you a webinar talking about a project in which we explore the genetic variation that is influencing the methylation in the brain and how this genetic variation can contribute to ADHD. DNA methylation is regulating gene expression, and environmental factors can modify this methylation. We focused on allele-specific methylation, or ASM, which is a common epigenetic mechanism in which the two alleles of a genetic variant can correlate with different levels of methylation. For instance, this allele correlates with higher levels of methylation, and this hypermethylation is often related to lower levels of expression. Whether this allele 2 is correlating with lower levels of methylation, and this is often related to higher levels of expression. Also, ADHD symptoms have been associated with changes in DNA methylation levels. So our hypothesis was that ASM may participate in ADHD susceptibility and that it may regulate also gene-environment interaction. So our objectives were to explore the contribution of ASM to ADHD through a case control study using the GWAS meta-analysis of the IPSYCH and the PGC, and then identify genes whose expression is modulated by these associated ASMs as new candidates for ADHD. So we started with the information of two previous studies performed in postmortem human samples in which they identified ASMs in different brain regions. When we combined all the information of these ASMs in the different brain regions and across all studies, we observed that there were like 34,000 SNPs, more or less, but that some redundancies were happening. For instance, there were more than one SNP correlating with the methylation levels of the same CpG site. The same SNP correlated with methylation levels in different CpG sites, and the same SNP correlated with methylation levels of the same CpG site in different burn areas. So what we performed was selection using different criteria, and then combining all this information, try to avoid redundancies. Selecting finally 3,900 SNPs, more or less, that we tested in the meta-analysis. So, well, I'm not going into much detail on this, but after multiple testing correction, we obtained 39 associated SNPs, and that correlated with methylation levels of 6 different CpG sites. These CpG sites lie within the promoter regions of 6 genes. And when we checked whether these SNPs also modulated the expression of these genes, we observed that for 4 of them, these SNPs were EQTLs for these genes, in which the risk alleles for ADHD showed higher levels of expression of ARTN, PADD1, and RPLP2 genes, whether the risk alleles for ADHD correlated with lower expression levels of C2 or AT2. Taking a look at the genetic context of the first CpG sites, these SNPs lie in the chromosome 1, in this genomic region, and these alleles correlated with methylation levels in cerebellum of these two CpG sites, being the ADHD risk alleles associated with lower levels of methylation of both of them. These CpG sites lie in a CpG island, and taking a look at the genes that were in this region, ERTN was one of them, and was the only one in which these CpG sites lied in the promoter region of ARTN. Also, when we took a look at the gene expression using the GTEx database, we observed that the risk alleles for ADHD correlated with higher levels of ARTN gene expression, which is exactly the opposite of methylation, which is what it was expected. And it was detected in cerebellar hemisphere, cerebellum, and nucleus accumbens, which is quite important for dopamine. Also, when we took a look at the epigenetic marks, this SNP here showed the highest number of epigenetic marks. I wanted to mention also that this genomic region was quite close to the genome-wide significant hit, the first hit effect of the GWAS meta-analysis. ARTN gene expression is more or less the same across all tissues. Well, it is involved in neuron survival, but it's quite interesting because it's involved in the survival of dopaminergic neurons, and since dopamine pathways are quite relevant for ADHD, this is a pretty good candidate. The second region involved more than 30 SNPs that correlated with methylation level in temporal cortex and frontal cortex for this CpG site that lied in the promoter region of C2R82. It's quite interesting because the risk alleles for ADHD showed decreased expression of this gene in several brain areas, some of them quite related to ADHD, and also those here more or less are the ones that had more epigenetic marks. C2R gene expression is quite restricted to brain areas and in high levels of expression, and well, so far its function is unknown. And the last region is this region in which these SNPs correlated with methylation levels in temporal cortex and caudal pons of this CpG site that lied in the promoter regions of PIDD that is expressed in this direction and RPLP2 that is expressed in the opposite direction. These SNPs were EQTLs increasing the expression of PIDD1 considering the risk alleles for ADHD, and also increasing the expression of RPLP2. And these two SNPs here were those ones showing the highest number of epigenetic marks. PIDD gene expression is more or less in brain is similar to other tissues, and it promotes apoptosis. And the other gene is not very much expressed in brain compared to other tissues, but it plays an important role in the elongation of protein synthesis. So far from the 4 candidate genes that we have, we will focus for future work in animal models of 2 of them, of ARTN, which was involved in survival of dopaminergic neurons, and C2R82, because although it's unknown, it was highly expressed in brain areas, and the risk alleles were EQTLs for many brain regions that were quite relevant for ADHD. And so this is it. I hope you enjoyed the presentation and it was not too much technical. Thanks so much. Bye.